Recall in the wall of arterioles, such as afferent arteriole, we do have smooth muscles. If the smooth muscles in the wall of arterioles contract, they narrow down, constrict the arteriole, so less blood flows into the arteriole. If the smooth muscles in the wall of arterioles stay relaxed, they open up, dilate the arteriole, so more blood flows into the arteriole. So we can say that the smooth muscles that we have in the wall of arterioles adjust blood flow through the arteriole. And when we focus on especially the afferent arteriole, we notice that the smooth muscles that we have in the wall of afferent arteriole are special. What makes these cells special is their sensitivity to detecting any changes in the blood pressure here and reacting to that change of blood pressure very rapidly. We name these specialized smooth muscle cells that we find them in the wall of afferent arteriole juxtaglomerular cells. Please note, sometimes we find juxtaglomerular cells in the wall of afferent arteriole too. However, in most cases, afferent arteriole is the place that we're looking for juxtaglomerular cells. This section of renal tubule, this is the ascending limb of nephron loop. When we look at the epithelial tissue, we notice that the epithelial tissue is one layer of cuboidal cells, except when we get to the wall that sits very close to the afferent arteriole. Here we can see that cells are columnar, they are tall, they are not cuboidal. And we name these tightly packed columnar cells that sit in the wall of ascending limb of nephron loop, sitting close to the afferent arteriole, macula densa cells. And we name both juxtaglomerular cells that we have in the wall of afferent arteriole and macula densa cells that we have in the wall of ascending limb of nephron loop, sitting close to the afferent arteriole, together juxtaglomerular apparatus. This apparatus has a significant role in regulating the rate of glomerular filtration. Recall, filtration would be pushing water and solute from glomerulus into the Bowman's capsule. And it is very important, no matter what happens to our systemic blood pressure, inside the nephron, the rate of glomerular filtration be regulated and adjusted very quickly. In this video, I'm going to discuss two different feedback systems that both adjust glomerular filtration rate. One of these feedback systems involves only juxtaglomerular cells. We call this myogenic mechanism. Imagine if blood pressure decreases inside the afferent arteriole. When juxtaglomerular cells do not detect that much pressure, they stay relaxed, and as a result, afferent arteriole stay dilated, blood flow increases inside the afferent arteriole. And please keep in mind, if blood flow increases means blood pressure in the glomerulus increases, and that increases the rate of glomerular filtration. However, if blood pressure increases inside the afferent arteriole and blood pushes the wall and stretches the wall, these juxtaglomerular cells detect that they are stretched, and within seconds, they quickly contract and constrict the afferent arteriole. So when afferent arteriole, because of the contraction of juxtaglomerular cells, narrows down, less blood flows into the glomerulus, which that leads to decreasing the rate of glomerular filtration. This mechanism that we name it myogenic mechanism, within seconds responds to changes in the blood pressure that flows into the afferent arteriole and quickly adjusts the rate of glomerular filtration. However, this is not the only way that kidneys adjust the rate of glomerular filtration. Sometimes it happens even though juxtaglomerular cells react and contract and narrow down the afferent arteriole, still glomerular filtration rate is too high. And keep in mind, when the rate of filtration is high, that simply means the filtrate is going to move too fast through the renal tubule. And the risk is when filtrate moves too quickly through the renal tubule, we do not have enough time to reabsorb the substances that we need. And some of those important substances are, for example, sodium ion, chloride ion, and water. So imagine filtrate is moving too quickly through the renal tubule, and inside the tubule, we still have lots of sodium ion, chloride ion, and water that we fail to reabsorb them. Macula densa cells detect the high concentration of sodium ion, chloride ion, and water, and then the communication between macula densa cells and juxtaglomerular cells start. The result of the feedback that macula densa cells send would be lowering the release of nitric oxide in the wall of afferent arteriole. Recall, nitric oxide is a vasodilator. So when the level of nitric oxide drops, juxtaglomerular cells can contract even more forcefully and constrict the afferent arteriole even more. When afferent arteriole constricts and narrows down more, the flow of blood decreases, and as a result, the rate of glomerular filtration decreases. And when we bring down glomerular filtration rate, that adjusts the flow of filtrate. So filtrate starts moving through the renal tubule with the appropriate pace that allows us to reabsorb the substances that we need. We name this feedback system tubuloglomerular feedback simply because first we detect the change using the macula densa cells, and the change would be too much sodium ion, chloride ion, and water inside the tubule, and then we have a feedback that eventually regulates the glomerular filtration rate. So we learn about two mechanisms, tubuloglomerular mechanism and myogenic mechanism. Both have the same goal. The goal is to adjust the rate of glomerular filtration. However, because in myogenic mechanism, the only cells that are involved are juxtaglomerular cells, this feedback system is much faster than tubuloglomerular feedback. Last but not least, I would like to remind you, as I explained in the video Renin, angiotensin, aldosterone, we know that when the systemic blood pressure drops, sympathetic nerves can stimulate the juxtaglomerular cells to release renin into the bloodstream. And we know that when renin is released into the blood, a sequence of events start that leads to the formation of a hormone named angiotensin 2 and the release of another hormone named aldosterone. And we know that those hormones, their impact would be increasing the systemic blood pressure. So there are multiple functions that we expect from juxtaglomerular cells. One would be the myogenic mechanism. The other one would be involvement of these cells in tubuloglomerular feedback. And also we know that these are the cells that release renin. Hope you find this helpful.